Hi everyone, this is Scott Reeves and this is an FSN 528 lecture. This is the second part of this talk on the regulation of fuel utilization in response to food intake. So I'm going to take off where I left off, and we talked about quite a few different things, and we were looking at how certain things are regulated on a cellular level. We talked about the AMP activated protein kinase and some of its effects. We talked about mTOR, and then we're going to now continue on. And one thing that we're going to look at is we need to think about what happens during fasting, but also what happens postprandial after a meal and how things basically shift. So again, we're going to look at hormones, but then we're also going to look at what happens inside the cell. So remember, we eat intermittently throughout the day, and one of the effects of that intermittent food intake is it changes the hormonal profile in our blood, and that can affect a lot of these metabolic pathways significantly. And if we review the anatomy of kind of the GI tract, remember that the pancreas is adjacent to the stomach and the liver, but the pancreas is part of the portal vein circulatory system. So the portal vein, the main blood circulatory component that removes water-soluble molecules from the gut, that portal vein actually goes right by the pancreas. So the pancreas is exposed to things like amino acids and carbohydrates almost immediately, and they hit the blood. And those pancreatic cells have the ability to respond, especially to amino acids, which is mostly leucine and glucose. And so when those molecules go into pancreatic cells, they can affect the release of hormones. Remember, the pancreas is a tissue or organ that's responsible for digestion, but also for the endocrine response to food intake. And if we look at the structure of the pancreas, we can see here that it has different portions that have acinar cells, which release things like enzymes and bicarbonate and some other molecules, but it also has an endocrine portion, which are kind of in these small little regions or structures called islets of Langerhans, and they're composed of mostly alpha cells and beta cells. Remember, the beta cells are releasing insulin, and the alpha cells are releasing glucagon. And when things like glucose and leucine go into a beta cell, they can increase the ATP production, and it's the increased ATP in a beta cell that stimulates calcium flux into the cell and then the subsequent release of insulin. It happens relatively quickly. Once those molecules enter into the beta cell or metabolize to produce ATP, and then the insulin can be released into the blood, and the effects can be relatively fast as well. Now, in fasting situations, the ATP levels in alpha cells are sensed, and in general, as blood glucose goes down, glucagon secretion goes up, and that glucagon is being released from the alpha cells into the blood. So any cells that have glucagon or insulin receptors can respond. And in general, when insulin is high, glucagon is low, and vice versa. Okay? So now, for example, how do we regulate glucose? And if we start off in the center here, we see that glucose homeostasis, which means in fasting scenarios, the blood glucose concentration is between 4 to 6 millimoles per liter, and that is 70 to 110, 70 to 100 milligrams per deciliter. When we consume carbohydrate, that glucose, some of that glucose enters into the pancreas, and the islet cells, in that case, receive the insulin, metabolize it, and that would cause insulin secretion to go up, and it would decrease glucagon synthesis. So when that insulin goes into the bloodstream, as insulin interacts with the receptors on muscle cells, it basically increases glucose uptake and utilization. When that insulin binds to receptors on liver cells, it also increases glucose utilization. It increases glycogen storage, and it can also increase lipogenesis. Okay? So basically, when that happens, blood glucose tends to go down. Remember, the liver takes up some glucose post consumption, but a lot of it goes to other tissues, including muscle tissue. And does the same. It can do the same thing in adipose tissue. It can increase glucose uptake there because of the GLUT4 mechanism is insulin responsive in both muscle and adipose tissue. Okay, now let's see here. Insulin also, if we look at the liver, decreases glucose production. So insulin decreases gluconeogenesis, and it decreases glycogenolysis. Okay, so it's storing glycogen, not breaking it down. Okay, so that helps maintain basically that glucose concentration in the 70 to 110. So in that case, it's helping decrease blood glucose to get back to that kind of level of homeostasis. Now, on the right side of this, when you look at starvation over here, so this is without food intake. We've looked at this before, but what happens in this case is, insulin is low in the pancreas, and glucagon secretion goes up from the pancreas, and that glucagon interacts with receptors on liver cells, and that decreases glucose utilization, decreases glycogenesis, decreases lipogenesis, but the glucagon increases glucose production and gluconeogenesis and glycogenolysis. And each of those, those are ways of increasing blood glucose concentrations. In this case, because of the low insulin, there is decreased glucose uptake and utilization in both muscle and adipose tissue. Okay, but the liver in this case is helping to maintain blood glucose within a certain baseline level. Okay, so the hormones change significantly. This is kind of what they look like. When you have, for example, an increase in blood glucose, we see up at the top, this postprandial changes in glucose, insulin, and glucagon. So after the meal, glucose goes up in the blood, and that is paralleled typically by an increase in insulin. And again, the higher the glucose, the higher the insulin. And then as the glucose is going into tissues, the blood glucose decreases, so that kind of feeds back and decreases insulin production or release. And if we look down at the bottom, we see that after a meal, glucagon typically goes down, and then as blood glucose goes down following the effects of insulin, then glucagon starts to come back up. Okay, we've gone through that a few times. So here's a little bit more on insulin signaling. I'm just going to add a couple more parts to this. So remember, almost every cell in the body has insulin receptors. Insulin binds to the top portion of this receptor, the alpha subunits. That causes a change in the beta subunits. The subunits are actually going through the membrane into the cell, and the receptor autophosphorylates, and tyrosine residues are phosphorylated, and that causes, for example, if we look down the right side of this, that phosphorylation of the insulin receptor stimulates the phosphorylation of insulin receptor substrate 1, which is bound to a protein called P85, and that P85 is bound to the phosphatidylinositol 3 kinase. It's also known as P110. That kinase, as its name implies, adds a phosphate to the number 3 carbon or phosphate of inositol. That converts phosphatidylinositol 4,5 bisphosphate to phosphatidylinositol 3,4,5, and that's a P3. So now that stimulates AKT, and that stimulates the phosphorylation of AKT. AKT is a molecule that can Sorry, I want to make sure I don't forget anything. Okay. The AKT is, let's see, what's the best place to look? The AKT actually leaves the plasma membrane, goes in the cytosol, and it does things like inhibits FOXO, which FOXO, one of the things that FOXO can do, it's a transcription factor that can stimulate proteolysis. So in this case, the insulin signaling and AKT phosphorylation is decreasing proteolysis. And remember the GSK3, that is an inhibitor of glycogen synthase. So AKT downregulates GSK3 so that glycogen synthase is activated. So in tissues that store glycogen, we have glycogenesis that is stimulated. And what else is happening here? So now let's look over towards the left side over here. Here we have the insulin receptor that's also stimulating an IRS molecule. In this case, it's interacting with a protein called GRAB2, and that's interacting with a protein called SOS, or son of sevenless. And those molecules are involved in myogenic pathways. In other words, there's a signal cascade. See that MEC1 and 2 and that ERK1 and 2? Those are mitogenic factors. So this part of the insulin signaling pathway stimulates mitogenesis, or basically mitosis or cell division. That's what's happening there. I think the other part, the outer part of it, see what it says, the CBL and the CAP and the TC10, that's part of the signaling pathway is actually stimulating GLUT4 translocation, which would be the insulin action there. So we have GLUT4 translocation for glucose uptake. We have mitogenesis that can be stimulated. And over here we have decreased proteolysis, increased glycogen synthase activity. Those are the main points on this one, all stimulated by insulin. Again, lots of different cells have insulin receptors. Some of them have other cell-specific processes like glycogenesis, but a lot of them respond in the same way to insulin. Okay, again, anabolic hormone. Now the opposite. Now when glucagon goes up, as we just saw, this is glucagon or catecholamine signaling. So glucagon is released, it's a peptide released by the pancreas, but we have epinephrine and norepinephrine, which are actually catecholamines. They interact, they all can interact with receptors that are work by the same mechanism. The glucagon receptors, they're going to have, liver cells have glucagon receptors, fat cells have glucagon receptors, muscle cells do not, but epinephrine and norepinephrine, they have receptors on 3 of those tissues: liver cells, muscle cells, and fat cells. Okay? But now when either one of those signaling molecules, either glucagon or epinephrine or norepinephrine, bind to the receptors, causes a structural change in the receptor so that that triggers a GTP to GTP exchange. In other words, GTP is converted to GTP in the alpha subunit of these G proteins. Okay. That exchange to GTP activates adenylate cyclase. Adenylate cyclase takes ATP and turns it into adenosine monophosphate, but it's in its cyclic form. So it cyclizes ATP, and now cyclic AMP stimulates protein kinase A. Okay? Now, again, these molecules are going to be functioning to provide energy in this scenario because, remember, this is a fasting or starvation scenario. So protein kinase A downregulates AMP activated kinase, which would make sense, right? AMP activated kinase tends to be upregulated in certain situations. In this situation, protein kinase A is inhibiting AMP kinase. Now, AMP kinase normally decreases lipolysis. So protein kinase A is inhibiting AMP K, so you lose the inhibition of lipolysis. So basically, lipolysis goes up. Okay, so it works through AMP by inhibiting that, but it also stimulates lipolysis directly. So it can increase activity of, this is hormone-sensitive lipase, which hydrolyzes the triglycerides in adipose tissue, and also paralipin, which is the protein that surrounds that triglyceride droplet. It modifies that so that hormone-sensitive lipase has more access to the triglyceride droplet. Okay, so that hydrolysis can generate fatty acids and glycerol. The fatty acids can go into the blood. They can be taken up by the liver. In this scenario, the liver can take those fatty acids, oxidize them, and generate ketones. So we have ketogenesis. The ketones can leave the liver, go to extrahepatic tissues like muscle, and be used for fatty acid beta oxidation. I'm trying to, it's like those ketones could also, not sure why they don't have this, be taken up by the brain and used as for beta oxidation in the brain as well. Oh, because that's fatty acid beta oxidation, sorry. So it's just muscle here. What else? Oh, the fatty acids. Now this is, again, kind of an indirect way, but those fatty acids that are released by adipose tissue can go into the blood. They can be taken up by the liver. The liver can take the fatty acids, convert them to triglycerides, and then put those triglycerides into VLDL, which then go into the blood, and those VLDL can interact with lipoprotein lipase on adjacent tissue next to muscle, and then they can be converted to fatty acids and used for beta oxidation. So essentially the fatty acids are going through the liver in that case, through VLDL, but still making it to muscle. Now let's backtrack here to the glycerol from the triglyceride that was liberated from adipose tissue. The glycerol can go to the liver, and in the liver that glycerol can be used for gluconeogenesis. Okay, so protein kinase A stimulates gluconeogenesis in the liver. It does it by inhibiting liver pyruvate kinase, and that's the last enzyme in glycolysis, which goes from phosphoenol pyruvate to pyruvate. So you inhibit that step, so PEP levels, phosphoenol pyruvate, stay elevated, and PEP can reverse back to glucose in the liver. Okay? Protein kinase A also decreases fructose 2,6-bisphosphate. That's the allosteric regulator of PFK1. So by decreasing fructose 2,6-bisphosphate, you're inhibiting glycolysis, and therefore that promotes gluconeogenesis. Okay? That also, protein kinase A also stimulates the cyclic AMP response element mediated transcription. And this response element is in the promoter of a few gluconeogenic enzymes like glucose 6-phosphatase. Okay? And so that allows gluconeogenesis enzymes to be elevated because of protein kinase A as well. Okay? So that gluconeogenesis provides glucose to the blood, so that glucagon is stimulating glucose release by gluconeogenesis, but the protein kinase A is also stimulating glycogenolysis in the liver, and that's providing glucose to the blood as well. Okay, so overall the main functions of each of these is providing glucose to the blood in that case. Okay, now let's kind of put some of this together in this slide. Two parts to this slide. Left side, high glucose, therefore elevated insulin. Right side, low glucose, elevated glucagon. So let's look at the left side first. Okay. And a lot of this is review, but it kind of puts it all together because the other slides weren't fun enough. Let's put it all together. High glucose and insulin. Insulin binds to its receptor, stimulates the phosphatidylinositol 3 kinase and AKT. And for example, let's look right here. Remember, those molecules decrease activity of FOXO, and FOXO gene expression is involved in things like gluconeogenesis, ketogenesis, VLDL synthesis, and proteolysis. So FOXO is inhibited by this part of the signaling pathway. Okay? Those molecules stimulate glucose uptake by stimulating GLUT4 translocation. They also inhibit the inhibitor of glycogen synthase. So they stimulate glycogen synthase regulated glycogenesis. So glycogenesis goes up in certain tissues. Okay? They activate the mTORC1 molecules. So amino acid uptake goes up. Protein synthesis goes up. Autophagy or degradation is inhibited. Okay. They also stimulate the sterol response element binding protein 1C, carbohydrate response element binding protein, and the liver X receptor. Each of these transcription factors promote lipogenic gene transcription. So insulin is basically stimulating lipogenesis. It's one of the things. As part of that process, acetyl-CoA is converted to malonyl-CoA. And remember, malonyl-CoA is substrate for fatty acid synthase. So the malonyl-CoA is providing carbons for lipogenesis. This is basically fatty acid synthesis. Those fatty acids can be converted to become part of triglycerides, which are packaged in VLDL in the liver, and those VLDL transport the triglycerides to storage in adipose tissue. So again, that high glucose and insulin can stimulate hepatic lipogenesis and storage in other tissues. Okay? That glucose, because of all of this, basically, glucose uptake goes up. Some of that glucose is used to make glycerol 3-phosphate through glycolysis, and that glycerol is now combined with the fatty acids that are from lipogenesis as part of triglyceride storage. So the glycerol is helping the triglycerides be made, and it's also, stimulating insulin, which is lipogenic. So it's providing carbons from glucose, and also the activity of the enzymes that are making the fatty acids to store triglycerides. Okay, let's look over here. Here is low glucose and glucagon. Now, when that happens, remember, cyclic AMP goes up, and that cyclic AMP can basically be converted to AMP. So in this case, increases AMP, and because of that, now you would have the AMP activated kinase. So you could take that whole other slide and basically just slide it right on here by the AMP activated kinase, right? And all those processes that are activated on that slide right, are also occurring here. Okay, let's look at this though. Oh, and this means that this phosphatidylinositol 3 kinase, AKT, are increasing this PDE3B, and this is an enzyme that decreases activation of the glucagon signaling. So in other words, if insulin is there, it decreases the glucagon signaling pathway. Okay. But if the glucagon pathway is activated, the cyclic AMP activates protein kinase A, and then the protein kinase A, we looked at this. Oops. Here. Stimulates glycogenolysis, provides glucose to the blood. Provides glucose, and then the glucose can go to the blood. Protein kinase A inhibits AMP kinase, and an AMP kinase is normally inhibiting lipolysis. So the protein kinase A is essentially inhibiting the inhibitor, so you get lipolysis. So the lipolysis in adipose tissue provides fatty acids and glycerol to the blood. The glycerol can go to the liver and be used for gluconeogenesis. The fatty acids can be used by other tissues like muscle for beta oxidation. And over here, that protein kinase A is decreasing. Here's fructose 2,6-bisphosphate inhibiting this kinase, this liver pyruvate kinase. And then this is the CREB that we talked about before. So the protein kinase A, once it's activated from glucagon signaling, is stimulating gluconeogenesis, stimulating or causing ketogenesis, causing VLDL synthesis, and MTP is what? Microsomal triglyceride transfer protein, something like that, an ApoB, so stimulating VLDL synthesis, and it's also stimulating proteolysis, and the proteolysis is providing amino acids that are basically used for gluconeogenesis in the liver. Okay, so kind of opposing end results in each of these processes. Some of them kind of work on similar molecules. Some of them use different molecules completely to regulate a specific process that's appropriate in either high glucose or low glucose. Okay, and then postprandial changes. How does kind of all of this kind of relate to what happens in the blood? So if we look at plasma concentrations of each of these, and we have glucose, this is time after breakfast. So say, for example, right before breakfast, glucose concentrations here. Post-breakfast, blood glucose goes up, and then of course once that happens, insulin's going to go up, and so the glucose is going to go down. And these are fuel sources, I guess I should say. And then we have total amino acids after breakfast. It's going to go up if it contained carbohydrate. I think they drew this to kind of a mixed meal with obviously carbohydrate, protein, and some fat. Triglycerides, which are shown where? Here's the triglycerides going up slightly after the meal for a few hours, and then they'll come back down. So this is basically, let's see, it would be chylomicrons and then potentially VLDL, where those triglycerides are. And then we have lactate, which goes up. The glucose goes up, it's metabolized, this causes an increase in lactate. And then free fatty acids basically are going down. Lipolysis is down because glucose is up. And then we have the ketone bodies are shown here. Oh, sorry, glycerol is here as well, and the ketone bodies, which are low because of food intake. Ketogenesis is low. Okay. So again, different tissues can use different molecules for energy. So we tend to have a mixture of a few different molecules, but most of those molecules are related to fats and carbohydrates being the major sources of energy. We have hormonal regulation that's an important part of all of this, and we have intracellular signaling pathways that respond to things like ATP levels, AMP levels, NADH levels, and NAD levels that are sensing cellular energy status and kind of responding in a more localized mechanism as well. So lots of different combinations of pathways affected by different supplies of energy. And again, this is happening 24/7. It should be a relatively intricate process that overall brings us back to that term we said before, and that is the metabolic flexibility, allowing us to adapt to whatever fuel source is available, and then responding when a fuel source is no longer available and another fuel source has to be used. Okay, so here's a fate of glucose from one meal, for example. This is a mixed meal, but we're talking about the glucose initially here. And this is if a person consumes a mixed meal that has 90 grams of glucose in it. Where does that glucose go? Well, some of that glucose goes to the liver. So if you consume 90 grams of about 20 grams of it is going to be used by the liver. And some of that is going to be stored as glycogen. Okay? Again, insulin will go up, so that stimulates glycogenesis. But if we look at the liver, lets quite a bit of that glucose go, and that allows about 15 to 20 grams of that glucose to reach the brain. And the brain will use oxidative phosphorylation and respiration. It'll generate CO2 and ATP from that. Okay? So in this case, that's the brain's main fuel source, is glucose. Okay? Some of that glucose will go to adipose tissue, and that's a relatively small amount. And but that glucose can go into adipose tissue, and for the most part, in this scenario, insulin would be elevated. That would stimulate lipogenesis within those fat cells, so you tend to have it stored as triglyceride. Now, some of that glucose goes into the muscle, and let's say it's 45 grams that can go into the muscle. About 25 grams of it is going to be stored as glycogen, and about 20 of it is going to be oxidized immediately. So for example, if you're exercising and you're going through a lot of muscle glycogen, and let's say for example you burn through 90 grams of glycogen in that muscle, you can't just consume 90 grams of glucose to replace it, because only part of it goes to muscle, and only about half of that is actually stored as glycogen. So if half of that actually is going towards muscle, half is stored as glycogen, half of it is oxidized. So pretty soon you go from 90 grams of glucose that you consume, only about 25 grams of it is stored in that muscle. So it takes a consistent high-level carbohydrate regimen to restore muscle glycogen in general. Okay? And of course, the amino acids in this case, quite a few of the amino acids from that mixed meal would go into the liver and be catabolized. Some of it would be used for protein synthesis in the liver. But a lot of those amino acids would be catabolized, and then some of those amino acids are used for protein synthesis in other tissues as well. And then the fat, most of the fat from that meal would go from chylomicrons and be stored in adipose tissue. Okay, so the liver, the carbohydrates has one of the more complex division of its contents between the tissues, because it's such an important energy source, and it tends to be limiting because we tend to use a lot of carbohydrate when available. Okay, and I think so that kind of summarizes we went through, but so I'm not really going to add any more, and I think that kind of wraps up this lecture, so I'm going to stop there, and I hope that was helpful. I hope you could hear it and everything was relatively clear.